On to our first session with Bart. So the first session is on microglial identity and expression profiles. And our first speaker is Bart Eggen from Groningen, and he's going to be talking about human microglia from a developmental and pathological perspective. Bart.
Okay, good. Good morning. Yes, my name is Bart Eggen. I'm in the Netherlands, Groningen. But before I start, I want to thank Guy and the organizers for giving me the opportunity to share some of the work that we did over the last 10 years or so. This is where I work, in University Medical Center Groningen. And I don't think I need to explain a lot about microglia. They're now 100 years ago, roughly, first reported by Pio del Rio Ortega. And many of us like these cells very much, and I guess that's why we're here, and I'm one of those people. So this is how they look like in a mouse brain, typical small soma, highly ramified processes, and over the last, I would say, 2-3 decades, there is many functions that are associated with microglia. I mean, they're CNS-resident macrophages, but they have a lot of functions that are tailored to their CNS environment. So they have a whole bunch of functions under homeostatic conditions, but also they are heavily implicated in a whole range of pathologies. And I will touch upon a few of these, and I will talk about published work, but also share unpublished data. So microglia are very heterogeneous in terms of functions, but also if you look at microglia themselves, you already can see that intrinsically these cells can be very diverse. So this is a dab stain of microglia in a mouse brain, and then you can already see that an individual microglia, depending which part of the cell resides in the gray and the white matter, the morphology is very different. So in white matter, they typically are much less complex and much less branched, and if they're in the gray matter, they're highly ramified. So even within a cell, you can find heterogeneity. But if you go to a disease, and this is work of Jade Stride as an example, but there's many, many more papers that I don't list. It's clear that microglia can adopt a whole range of phenotypes and morphologies in the context of disease, aging, sepsis, etc. And what we don't know in the field is if all these different morphological states translate into functional states, and how many functional microglia states do exist. This is something we will also discuss later in the week. I think at that moment the field does not really know. And the approach that I use in the lab is that I use a lot of omics approaches. So I used single cell sequencing. I started off by bulk, and then I do single cell, and I do a lot of spatial omics, trying to identify microglial states, transcriptional states, and I also do protein work to identify if microglia change and how they change in the context to pathology. And the hits that we find, we then test in cell models or mice or organoids, etc. So I'll share a few stories with you, and I'll start in the human brain. That's basically where I start all my work. I start in the human brain tissue. That's the starting point, trying to identify microglia states or subsets and then fish out what they do. So a trip down memory lane, a few years ago, we isolated microglia from fresh post-mortem tissue from roughly 100 cases or so. And then what we typically do is that we homogenize the tissue, we fact sort out microglia using cell surface markers, and then we do RNA sequencing. So this was still in the bulk era, so this is bulk RNA-seq of microglia. And what we found is not so surprising in the sense that they are indeed the CNS-resident macrophages. They express a lot of receptors on their membranes that they need for their sensing activities. So they have a lot of sensome receptors, they have a lot of cytokine genes that are being expressed. So this is all typical. And what we knew from the mouse work, but what was interesting to us is that if we look at the genes that change across the human lifespan, because we isolated microglia from donors ranging from 30 years of age to 100 plus, if we then isolate the genes that change in microglia in relation to age, we see that there's a whole bunch of genes dysregulated, and these primarily land into genes that encode for components of the actin cytoskeleton machinery, and also a lot of genes that encode sensome, or cell adhesion molecules. And this is fundamentally different from what you see in mice. If you look at young and old mice, it's typically inflammation or phagocytosis-associated genes, but this is not the case in humans. The genes that change in microglia in relation to aging are different from what we found in mice, but this is published. So then we moved to the other end of the spectrum, and we looked in human fetal brain development. So we had a very simple question: how do microglia mature or develop in the human fetal brain? When we started this work, not much was known about humans. What was known in was mouse data, where mice were processed at different ages during development, using a range of technologies. Could be a single cell sequencing like here, also epigenetics. And basically this is work from the lab of Ido Amit, and they showed that microglia in the mouse have different phases of development. And early in development, they're characterized by being very, I lost my mouse, oh here it is. They're characterized by being mitotically very active, which I think reflects the fact that the mouse is developing, the neural epithelium is expanding, so you need more cells. And then around birth in mice, they are very, express a lot of genes that are implicated in synapse pruning, so basically network refinement. And then at the end, they adopt a typical microglia surveying phenotype as we know it. And that work has then since then been complemented by work from Beth Stevens' lab, the Hammond paper in Immunity, where they did a similar study. And a few years, last year and the year before, work from the lab of Diego Nicola showed more work on how microglia develop in the human brain, but that was last year and the year before. So how microglia expand and colonize the human CNS and how they change over a human lifespan. But we really focused at the earliest events, so we started this work, I would say, 5 years ago, and what we did is that we isolated human CNS samples from human embryos. It was fresh tissue, and then we would isolate from the CNS samples and the spinal cords. We would isolate microglia, and it's plate-based single-cell sequencing and also epigenetics, which I won't talk about. And what you get then is a UMAP. So I will show you more UMAPs later in my talk, but what is a UMAP? So here each dot is a cell, so we're seeing a single-cell sequence, 20,000 microglia, and then you put your 20,000 microglia transcriptomes into an algorithm, and it looks for populations of cells that have similar gene expression profiles, and that's what you see here. So each cluster is a population of microglia that has a similar gene expression profile. This is what these clusters are. And then what we did is that we did several things. I won't show you the data, but cluster 6, for instance, is a cluster that is present at all ages, in all embryos, and these are proliferative microglia, so they express a lot of genes involved in chromosome segregation. But if you look at, we were looking at clusters that change across the gestational weeks of these embryos. So what you can do is you can calculate. From each of these, for instance, clusters 7 and 8 at the top, from each cell we know from which embryo it came, and from each embryo we know how old it was. So what we can do is we can calculate for each cluster from which embryos were they derived. Is there a difference in proportionality across lifespan? And what you see here is that microglia in these top clusters are primarily derived from very young embryos. And the next cluster over, these two, are primarily derived from embryos that were a little bit older. So there seems to be a shift in microglia states across development. And if you then look at the genes that were abundantly expressed in these 4 clusters, they're quite reminiscent of the genes that were reported by the Amit group to be induced in a response to amyloid. This DAM signature, we will discuss later in the week all the different flavors we have, DAMs, ARMs, PIGs, MGNDs, you name it. And it's also reminiscent of a gene expression signature we published in 2015 when we basically did a meta-analysis of microglia in mice in a whole series of CNS diseases. And then we saw that there's this sort of constant gene signature that is increased in these cells, and this is something that we also find very early in human fetal development. So these cells express genes that later in life, under neuropathological conditions, are reactivated. If you look a little bit further down, so these clusters 1 and 4, they are mainly derived from older embryos. So these are, and if you look at the genes that are abundantly expressed in these microglia, they are the genes that we know from our adult human work. So homeostatic microglia, sensing, that's what these clusters primarily express. So that suggests already in the first trimester of human development that in microglia there is some maturation occurring. You can do different things with these UMAPs. So this is basically plotting how transcriptional heterogeneity takes place. but you can also superimpose the age of the embryos from which each cell was derived. And then you can see that there's a gradient, so the youngest embryos are in reddish at the top, and then the more turquoise is an 18-week-old embryo, and then you can see that there is a pattern. And we analyzed that using computational tricks, and indeed we can calculate how microglia mature in the first trimester of human pregnancy. So in a nutshell, what we showed is that early in development, microglia in the first, this is basically 9-week-old human embryos. Microglia are quite activated, or they express a lot of genes involved in phagocytosis, but already in the first trimester, in a span of 10 weeks, they change their gene expression profile and adopt a phenotype which is fairly reminiscent of what we find in adult humans. So they express a lot of homeostatic genes, and that might be an important driver of CNS development, but we can discuss that later. So now something else. So this is development, and now a little bit of pathology. So I was interested to look at frontotemporal dementia. So what is frontotemporal dementia? It's the most common form of dementia after Alzheimer's disease. And it's caused by, it can be familial, so there can be mutations in genes, or they can be sporadic. Roughly 1/3 of the cases carry autosomal dominant gene mutation, and the genes are listed at the top. These are progranulin, C9orf, or MAPTau. These are the 3 main autosomal dominant mutations that can cause FTD. And as the name indicates, it's primarily the frontal and temporal cortex that's affected. So here you see in stain, so you see here immunohistochemistry for NOI-N, which is a neuronal marker. And then you see the frontal, temporal, and occipital cortex of cases and controls. And I hope you can appreciate that if you look at the frontal cortex, there's hardly any NOI-N labeling left. So that means that all the neurons are pretty much lost. And you see that in the occipital cortex at the bottom here, you see that is hardly the case. So in this study, what we did is that we focused on patients that carried progranulin mutation, because that's a very severe phenotype. And we were interested in what's going on in microglia, because if you knock out progranulin in mice, they develop microgliosis and neuronal loss. So that's why I was involved as a microglia biologist. So what did we do? We isolated the frontal, temporal, and occipital cortex samples. We took them from frozen specimens. We isolated the nuclei. We labeled the nuclei with antibodies that discriminates between oligodendrocytic lineage with OLIG2, NOI-N to get rid of all the neurons, and then we collected the double negative fraction because we were interested in microglia, which would be in this quadrant here. And that we subjected to single-cell sequencing, and we sequenced 500,000 nuclei or so, and then 120,000 landed in microglia, I think, over 100K or so. So this is then a UMAP with all the different cell types from which these neurons were derived, they cluster together. So the first thing we did is that we looked at microglia. So this is the microglia UMAP. So it's a subcluster of the total data set we got. And we find all kinds of clusters. And there was one that stood out, cluster that you cannot, so cluster 1, but I'll enlarge it. So cluster 1, so this is frontal cortex, temporal, and occipital cortex. And you can see that the solid bars are controls and the open bars are cases. You can see that this cluster 1 is highly enriched in the samples that come from progranulin carriers. So we wondered, what is this cluster 1 microglia, and how is it different from homeostatic microglia that are in cluster 0? And the long story is not very much, because there's very few genes that are differentially expressed between cluster 0, which is controls or homeostatics, and cluster 1. Even with very lenient log-fold changes, we could not find very many genes, so we really don't know what is going on. But we do know is that the changes in gene activity in these microglia are very subtle. And then we went back to the tissue and immunostained it, and we calculated, so we looked at morphology, we looked at the number of cells per surface, we looked at the area covered by IBA1 signal, and none of it was different between cases and controls. So basically, we were a little bit disappointed because we were looking at microglia changes, but it also is good because it shows you that an unbiased single nuc RNA-seq approach can give you surprising results, and at the end it all worked out, I'll promise. So what we did then is that we used a cell interaction prediction tool. So this is becoming more and more popular. So what you do is that you take your single nuc RNA data, There's now databases where ligands and proposed receptors are leveraged. And then you can ask in your data set, is there between certain cell types a predicted changed interaction in terms, do some cells over or under express ligands and receptors? So that would predict if interactions between certain cell types is changed. There's no proof, but it gives you a handle on in your data which cell types would be worthwhile to pursue. And that's what you see here. So on the left, you see all the senders, meaning the ligands. At the bottom, you see the receivers. These are the targets. And if there is a changed sender-receiver interaction, if it's increased in FTD, the boxes are red. And if it's decreased in FTD, it's blue. So you see there's this whole block of red interactions. And these are all cells of the neurovasculature. These are astrocytes, fibroblasts, mesenchymal cells, endothelial cells. These are all a member of the neurovasculature. So that's what we looked into next. The astros are very changed. Again, immunohistochemistry across all the different brain regions we analyzed, and then cases versus controls. So you can see this is a GFAP stain. There's extensive astrogliosis, as you can see, in the frontal cortex, but not occipital. And if you quantify that, we see more astros and a lot more and severe astrogliosis. When we subcluster the data, apparently it doesn't like subclustering. Oh, yes. So when we subcluster the microglia, we see two clusters, clusters 4 and 5, that are highly enriched in FTD cases. And then we looked at the genes that are that are enriched in these clusters. That's the genes. And one stood out to us, which is WDR49, which nothing is known about. It's a gene that has 1 or 2 papers. And we knew somebody at the Human Protein Atlas in the Karolinska, and he had antibodies against this protein. So we were very interested. So when we label it, we see that it labels astrocytes in a very particular manner. These small spheres of astros that express WDR49. It's exclusive for the frontal cortex, and we've looked now in many other neuropathological cases, and it seems to be a new biomarker for diseased astrocytes and CNS diseases. We are interested in what this protein is doing. We don't know yet, but it's exclusive for, in this case, FTD in the frontal cortex. You don't see any label for this protein in the different regions. Okay, back to the neurovasculature. I want to have to give you a little bit of context. So the neurovasculature, arterial blood comes in from your heart, it goes into your arteries, arterioles, then it enters the capillary bed where you have exchange of nutrients and oxygen and CO2. Then it collects in the venous part and goes back to your heart. And these different parts of the neurovasculature you can identify because all these cells that are in there have their own gene expression signature. So you can, based on gene activity, discriminate between endothelial cells, your arterial, capillary, and venous part. And you can also discriminate between smooth muscle cells and your arterial part, your venous part. And the ones in your capillary are called pericytes. So you can tease them apart. A paper came out in, in this paper in Nature. This is mouse work, and there they annotated all the different vasculature cells, and if they were arterial, capillary, or venous, and we superimposed that on our human data. And it worked. So this is a UMAP of, I'll only show you endothelial cells. So this is a UMAP of endothelial cells, and we could identify the endothelial cells derived from the arterial end of the structure, the capillary cells, which is most of the nuclei, which makes sense because the capillary is the largest part of your neurovasculature. And these are nuclei coming from the venous endothelial cells. And then we superimposed here these gene signatures, so you can see arterial nuclei are here, capillaries are in the middle, and at the end you have your venous part of your endothelial cells. And then you can calculate a trajectory. So basically what you do is you order your nuclei along a fictive timeline, and that timeline follows the arterial, capillary, venous structure of the neurovasculature. And then what we did then is that we calculated the number of nuclei along this trajectory in cases versus controls. And that's what you see. So if you focus first at the bottom, the solid lines are controls, and the dotted lines are FTD progranulin cases, and you can see that these lines overlap, meaning that the peak in nuclei is here, meaning that most nuclei are derived from the capillary bed, and you can see that there's no change between cases and controls. But if you look at the frontal cortex, you see that the dashed line is left shifted, meaning that there's more, that the nuclei in the endothelial compartment have a more arterial signature. Might reflect a perfusion problem, that your arterial bed is expanded in FTD carriers. Anyway, so the bottom line here is that we started this project as microglia biologists, being interested in how microglia might drive FTD progranulin. But the good thing is that we figured out that it's the neurovasculature. So it's good, because as a microglia biologist, I learned something about blood vessels. So I would say it's a win-win. How am I doing for time? Oh yeah, I'm good. A last bit I want to share with you is, again, a different disease. This is all unpublished work. It's multiple sclerosis. So a brief introduction. So multiple sclerosis is a relatively common autoimmune disease where your immune system damages the myelin sheet around your axons, and that leads to progressive neuronal loss. On the left, you see how the typical disease course of MS. So most patients, it's diagnosed typically in your 20s, and then most patients develop these bouts of inflammation, which are then followed by a phase of recovery. So this is the phase of the disease which is called relapsing remitting. And this phase of the disease is also quite treatable with anti-inflammatory drugs. That's or B-cell depletion. That's basically the approach. And then at some point, patients convert from a relapsing remitting to a progressive phase of the disease. Then you don't have these shoops or these bouts of inflammation. You have a constant deterioration, and that is accompanied by a reduction in brain volume and increased disease burden. So on the right, you see basically what happens. This is normal myelination of an axon, and then in MS, you lose your oligodendrocytes. Your axons become barren, and at the end of the day, you lose your neurons, leading to disability. Many cell types are implicated in this disease, and microglia are also. And we were interested in how microglia are involved. A little bit more background. When you suffer from MS, these lesions emerge in your brain. They're demyelinated, but lesions can arise in so there's different types of lesions. For instance, there's an active lesion, and the idea is that this is a relatively new lesion, where in the core of the lesion you have a lot of inflammatory cells. Demyelination is ongoing, and this is a process where the idea is that this is one of the first stages of lesion development, and that's where myelin is being damaged and removed. Then you have chronic inactive lesions, or mixed lesions. The core is relatively hypocellular. There's not a lot of inflammation going on in the core, but if you look at the rims of these lesions, you see a lot of ongoing demyelination and inflammatory activity, and there's a lot of microglia implicated. And there's two other lesion types, inactive. So in these lesions, you see a demyelinated core. There's very little immune activity. It seems that might be the end stage or the end result, but there's no repair taking place. But you also can find remyelinated lesions. They're called shadow plaques, and this is where the myelin has been repaired, but not to the extent as in wild-type tissue, let's say, in the control tissue. So you can still see them, that's why they're shadow plaques. But here successful remyelination took place. RIMs are reported about these active and mixed active and active lesions. They're known, and there's a lot of focus on this because that's most likely where the action is. That's where the lesions expand or shrink, and where ongoing demyelination is happening, so there's a lot of focus on the processes that drive the lesion expansion. So a lot of work has been focused on this, because this is a cartoon of the lesion rim, because at the rim you have, especially in active lesion, in these mixed lesions, also in the active lesions, you have a lot of microglial activity, monocytes or macrophages are being recruited, so you have expansion of the lesion. And this is work from Martina Absinta that she published 2 years ago. You can also follow some of these lesions in MRI in patients, and then she could show that these mixed lesions, you can detect the rim because the rim is loaded with iron, and she saw that these rims were expanding, meaning that over time these rims are growing. And another important factor is that there's evidence that the number of these mixed lesions a patient has correlates with disability. So they're important to understand what these lesions are doing and how do they expand. Okay, in a nutshell, this is, I'm not going to go into the details. In the rim, a lot is happening. So myelin is being damaged, myelin is being repaired, OPCs are being recruited. They need to differentiate into oligodendrocytes, they need to myelinate. Macrophages are involved, in the one hand, in removing myelin debris. On the other hand, they are important in microglia important in OPC recruitment. So there's all kinds of mixed processes going on, and you have many different microglia states that are important in this process. Labs have tried to address this by using spatial methods, and these include laser capture or punch biopsies or manual dissections to cut out rims to figure out which processes are perturbed in the rim. So I think 4 years now ago I started doing spatial omics, and this is and we started off with Cartana, then we switched to Visium, and basically what you do is on the top here, what you see, this is an MS lesion, an H&E staining, and here you see the lesion, which is the myelin depleted, and we cut a section, we place it on a glass slide. These glass slides have probes, these probes capture the RNA, and then you can sequence that, and all these probes have a address, a zip code basically, so you can place back your RNA in your tissue. And then this is what you get. So this is, for instance, a spatial plot for PLP. So this is PLP RNA that you're looking at, and you see that PLP RNA is depleted right where the lesion is. So that's how you correlate the data. So Visium is, you look at the whole transcriptome. The only drawback is that the spatial spots are large. There's a new version coming out which is smaller, but these are still relatively large, 55 microns. So that means it's not single cell resolution, it's 1 to 20 cells in each spot. To tackle that, what we also did is that we designed a 250 probe gene panel based on the Visium data, and we used these probes to validate our data, and in situ sequencing is single cell resolution. So we could go back to the tissue, do in situ sequencing, and check in which cell types these genes were dysregulated. So a few spatial spots to illustrate that this works. So CWM is control white matter, so this is white matter from control donors. Normal appearing white is white matter from a person that suffered from MS, but there's no apparent lesion. And these are the lesion types I explained, and then you can see that, for instance, the APOC1 gene clearly labels active lesions and the rims. This is osteopontin. It also labels the rims around mixed lesions, and the same is true for osteopontin, and you don't see any of these signals left when the lesion is remyelinated. And if you then take these same probes and we do in situ sequencing, we see the same. We see that osteopontin here and APOC1 label the rims of these lesions in active and mixed lesions. So that works. So the paper is in revision, so when it's out, the data will be accessible, so you can go into a web application, type in your favorite genes, or combination of genes, plot them, and then your spatial plots will emerge, and you can the violins, and you can download the data and check where your favorite gene is expressed in MS lesions and when. But that's hopefully when we convince the reviewers this is worthwhile sharing with the community, you can knock yourselves out on this website. Anyway, so I think it's something where we spent a lot of effort in is that you can generate these spots, you can generate these gene expression profiles, you can do cluster, you can look at gene expression differences, but we really were interested in can we use these data to predict how lesions evolve? Can we do that? So can we build trajectories using these spatial data and calculate how lesion evolution takes place in MS? And this is a bioinformatic trick, I understand, but maybe it is worthwhile. So what we did, this lecture is like death by UMAP, I'm sorry. But on the left, you see a UMAP again, but each dot is not a cell, each dot is now a spatial spot. Okay, so and each spot has multiple cells on it. But anyway, still, you can calculate, you can still do calculations on your spatial spots. You can still order them and see if there's patterns in your gene activity along these spots. And that's what we did. You see that in the middle. So that is a pseudotime along the spatial spots, and you see two trajectories, one going here to the right or top, and here one to the bottom. So basically the data split, and we look at, and I'll talk you through one of these trajectories to see what's going on and can we understand what drives this trajectory. So there's several layers of information that you can superimpose on these data. For instance, we know there's a whole bunch of cell types implicated in MS lesions, oligodendrocytes, microglia, that's the first one. These are mature oligodendrocytes. AIMS, astrocytes inflamed in MS, reported by Absinta. Microglia inflamed in MS and foamy microglia. A microglia subset that we identified by single-cell sequencing of human MS lesions. Plasma cells that secrete antibodies, fibroblasts, vasculature, and they all have gene sets that we know, and we superimpose them on our trajectory. And then you can see that oligos are enriched at the beginning of the trajectory, AIMS and MIMS, and these microglia all a little bit in the middle, and the plasma cells a bit to the end, and all the way at the end of the trajectory, that's where you find your vasculature and your fibroblasts. So that suggests that sort of at the beginning, the process from the beginning of the trajectory to the middle is a demyelination. You lose your oligos and you acquire activated astrocytes and microglia. We see that, and we will discuss the terminology, but anyway, that's what we see. So you see an enrichment of microglia and astrocytes that we know are changed in the context of MS. The next thing we see is that we see a lot of immunoglobulins, so the immune component, and it ends with fibroblasts and vasculature, which is known. We know that the core of these lesions is fibrotic and highly vascularized. We know that. So we sort of can reconstruct how lesions evolve. But we have another layer of information. We know from each spot for which sample it came, from which lesion it came, and where it was. So we can also superimpose not gene activity data, but we also can superimpose from which experimental group was each spot derived. And then you can see that the spots coming from normal appearing white matter tissues were predominant present at the beginning of the trajectory. But if you look at the rims, where all these microglia and astrocytes are enriched, they locate to the middle of the trajectory. That's exactly where we expect them to be. Activated microglia and astrocytes should be in the rim of these lesions, and that's where they are. And at the end, if you look at the core of an inactive or a mixed lesion, that's where you find your fibroblasts and your vasculature. So we sort of, by doing this computational organization of your spatial data, you can sort of reconstruct how lesions evolve and how they sort of evolve into each other. And then you can sort of predict how lesions evolve in MS. So it starts off with normal appearing white matter. And then we don't know why, but then a lesion emerges, and that it first starts off as an active lesion. And that lesion can be remyelinated. It requires the recruitment of OPCs and oligodendrocyte maturation, then you get successful remyelination. But the data also suggests that if an active lesion is not repaired and it evolves into a mixed lesion, there's two strategies, or two options, I would say. One is that it can end up being an active lesion, which has no activity, a demyelinated core, and basically that it will not be repaired, it's an end stage. Or it can stay a mixed lesion, and that they can also be very stable, as I showed you in the MRI data. They've been seen for 7-8 years in a patient. They could be a sort of semi-endpoint. And our data suggests that in order to remyelinate, you need to go through an active lesion state. And we hope and we think that our data suggests that if you're an inactive lesion, that you can go back to an active state. But we are very interested in the biological processes that drive all these different bifurcations. And we know that microglia are critically implicated in the first transition, the transition from normal appearing white to an active lesion. That is clear. But I must also say that Visium is not particularly good at picking up microglia because they're relatively small, relatively RNA low. So we have now different machines that have a higher resolution, higher sensitivity. So next time around I can tell you a bit more how microglia change NMS. All right, so this is the group. These pay the bills. And these are former PhD students and collaborators. So the people at Erasmus MC were critically important for the FTD project, because John and Haro and Lucia are neuropathologists and neurologists that treat these patients. In Amsterdam UMC, we collaborated for all these validations that I showed you some of. And at the Brainbank in Paris, they were key because we did a replication study in FTD cases from France. And we saw the exact same thing as in our Dutch cohort. And with that, well, if you're a postdoc and you like omics, drop me a mail, and we have openings. And otherwise, I want to thank you very much for your attention.
Thanks very much, Bart. That's a fantastic start to our meeting. So, questions. Does anybody have any questions? And if you have a question, can you use the microphones because we have participants online?
Thanks very much. That really was a fantastic talk. You took us to UMAP heaven. So I have two questions. First of them, it's in your developmental story. You showed nicely you have the dam-like cells, and then later on in development, you have more like a homeostatic state. Would you care to comment on whether it's those activated cells that become deactivated later in development, or do those cells die off and they're replaced by other cells to create this homeostatic cluster?
Yeah, so we don't know for sure, but I think that our computational analyses suggest that these cells intrinsically change. So it's not that they die off and a new batch becomes homeostatic, but I think it's a normal process, and these cells are still transcriptionally flexible, I would say, so that they mature. And it's also not a black and white, it is a gradual shift from more phagocytic to being more homeostatic.
Thanks. And the second question relates to the FTD work, so with the vascular changes that you saw. Would you care to comment on whether the vascular changes were secondary to disease or primary?
Yeah, it's a very good one. We don't know. The short is, I don't know. But we're looking at other FTD cases with different genetic drivers, checking if we, again, have neurovascular issues. It could also be that the vasculature, so it is also clear that the vasculature in the frontal and occipital cortices are different. If you look at homeostatic, it's different. So that might be an underlying factor, because we don't know why is only the frontal cortex affected and occipital not. We don't know. So it could be an interplay, but what comes first, I have no idea. And what makes it a bit complicated is that the phenotype in knockout mice does not recapitulate human disease. So it's not so easy to test. But it's a good question. I don't know.
Other questions? There's a microphone coming.
Hi, I'm not sure if you've done this, but do you know anything about what happens in primary progressive in terms of the comparative trajectory that you're stating for secondary progressive MS?
Yeah, no, we haven't done that. That was something we should do. We should check if there's a difference between relapsing remitting and primary progressives. Yeah. But most of the samples that we have are from people from the brain bank that at the end donated their brain. So most of them are secondary progressives, I would say.
And what about, can you comment on your trajectories for human versus an MS model, an animal model? Because there's a lot of debate about whether MS models really do mirror what you see in humans.
So we haven't done it, but just a paper came out by Gonzalo Branco Costello, who did this, who did EAE, and he processed spinal cords at different time points, and he could show that at least some of the things that we see was also happening in his EAE model. The timing and when the cells are implicated and where are they located. So, but yeah, of course there are differences, but I think still what he shows is that if you're asked the right questions, these models can address important questions.
Yeah, that was an excellent presentation. Just one question. Many years ago it suggested that microglia may be an initiating factor in MS. So the question is, does any of your data support the idea that the microglia may be actually firing the process up, or are they simply playing roles subsequent to demyelination?
Yeah, I think the most recent GWAS paper sort of indicates that some of the risk genes that land on microglia might predict disease severity and how fast it progresses. I'm not sure that there's a lot of evidence that the initial, well, first of all, we don't know what initiates MS. It's EBV, it is HLA locus are the main hits. So there's not a lot of compelling evidence that in a population-wise, microglia are driving it, but there's now good evidence, and I think that makes sense, that microglia risk genes might actually drive how the disease develops, how fast it goes, and the same is true for other diseases. I don't think microglia are the drivers of every CNS disease we have, but But I do think that genetic risk might say something about the speed that you develop, how fast do you deteriorate. I think that's what a lot of microglia risk genes do.
Thank you for your compelling presentation. I have a question about, so most of your results in multiple sclerosis are based on white matter lesions. So I was wondering, did you try to see what happens, for example, with subpeel lesion, which is something we use for the motor cortex, for example, to see if we have multiple sclerosis. So I wanted to know first, did you try to see what happens with the lesions in the gray matter? And did you try to see differences or heterogeneity with your spatial transcriptomics in motor cortex, for example, or spinal cord?
Yeah, so we have done gray matter lesions. We looked at subpiles and leukocorticals. We are still analyzing the data. We're increasing the N. What makes gray difficult is that differences between the layers in the cortex is driving a lot of the gene expression signatures that you get. So computationally, it's quite complex. We're working on it. Spinal cords we haven't done, would be fantastic. So the long story is we're on the same line, not done yet.
Okay, thank you.
Okay, I think we have to move on now, but I mean, you don't happen to know whether there's any online questions? No. Great. So our next, thanks very much.
Yeah, sure.
Bart.